All right, we sharing that. All right. So the reason we're here today is talk about blood. All right. Now, the hematologic system just refers to a division of the cardiovascular system, which is the blood that's carried by the blood vessels and pumped by the heart. Okay. And one of the things to realize about the circulatory system in general is it's actually a solution to a problem in evolution. All right. Back in the day, when life first appeared on the planet, there were single-celled organisms that had to use diffusion and active transport to get what they needed from the environment in order to survive. Basically, the world was covered pole to pole in water, and the environment was fairly uniform. What happened as the planet began to change is that new environments began to crop up. All right. Parts of the world got colder, parts of the world got warmer, parts got drier, parts got saltier, and so on. And with these new environments cropping up, life had to fill these niches. Okay. And so what happened was that you had to either adapt or die. Right. And that's the mechanism of evolution and natural selection that runs on mutation in order to pick out traits that suit an organism for a particular lifestyle in the environment. So, one of the first things that happened going from single celled organisms to a different architecture is the formation of something called biofilms. And y'all probably heard a little bit about this when you took micro. All right. But a biofilm is basically a matrix. And inside the matrix are cells, bacterial cells in this case, and they form a community, right? So we go from single-celled organism. Lord, that's terrible. Single cell to biofilm. Okay. And then eventually what we get to is first multicellular organism. With actual body systems and an increase in size. And so if you can look at the biofilm, what you see is actual division of labor among the bacteria that live in the matrix, such that there are going to be teams of bacteria that are good at making the matrix, teams of bacteria that are good at acquiring nutrients, teams of bacteria that are good at defending against attack. Okay. And they actually all communicate through something called nanowires. And biofilms are still around today. Okay. That's the plaque on your teeth in the morning when you wake up and brush, right? That is actually a biofilm. Okay. So we can example plaque. But once we get to a multicellular organism, we have a problem. And the problem is we need a delivery system for nutrients and oxygen and removal of waste products for cells in tissues that are not near the respiratory system and the digestive system. Okay. And it's the circulatory system that provides that. All right. You can think of it as the body's pizza delivery and waste removal service, basically. Okay. And it consists of the blood vessels, and the heart that pumps it. And today we're concerning ourselves primarily with just the blood. All right. So let's look first at blood, a brief overview, and then we'll talk more about it.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue, they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
So different formed elements in a liquid matrix. So bottom line is that blood is connective tissue. It is liquid connective tissue. Y'all remember from A and P that connective tissue always features cells embedded in a matrix with lots of matrix between them. That's the first thing that jumps out when you look at it under a microscope. In the case of blood, the matrix is something called plasma, all right? And the cells are the erythrocytes, the leukocytes, and the thrombocytes, which are the platelets, okay? Other connective tissues have different types of matrices. Bone, for instance, has a matrix made of calcium phosphate and collagen mostly, right? Connective tissue proper has a matrix that's made up of elastin and keratin and collagen, okay? But this is a liquid connective tissue. Now, the other thing that's distributed by the blood, in addition to the enzymes, the hormones, the immune components, the nutrients, the oxygen, and removal of the waste products, is heat. Okay, it is a heat distribution mechanism as well, because the bulk of plasma is made up of water, and y'all know that water has a high heat of fusion and heat of vaporization, which means that it can take in or give off a lot of heat without changing temperature very much. And so this helps stabilize the body temperature, right? So the chief functions are listed there, right? Delivery of substances needed for metabolism, waste removal, defense against disease and injury, maintenance of acid-base balance, okay? Blood's 92% water, 8% solutes. What are those solutes? Waste products, proteins, nutrients, hormones, enzymes, okay? The bulk of the plasma proteins are made by the liver, all right? This is one of the reasons that people with liver damage have terrible problems with the normal function of blood, all right? You have about 5.5 liters in the body. Plasma is approximately, let's call it half the blood volume. The liquid portion that contains organic and inorganic elements is the plasma. Your hematocrit is defined as the number of packed red cells that you get if you take whole blood and you spin it in a centrifuge, okay? So you would just basically take a crit tube, which is a hollow glass tube, you touch it to a drop of blood, that drop of blood would wick up into the tube. You plug both ends with clay. You spin it in a special centrifuge. It'll spin those little tubes. And then what you'll see are packed red cells on the bottom, and then a buffy coat in the middle, and then plasma on the top. And the percentage of whole blood that's packed red cells is your crit, okay? The plasma proteins, which are made primarily by the liver, are listed here, right? Albumins, which function both as a buffer but also as a way for the blood to hold on to its fluid volume by osmosis. Globulins, which are carrier proteins for lipid-soluble substances, right? Lipid-soluble stuff does not dissolve in water, so it needs a way to dissolve, and that's what those transport globulins are for. For instance, if you have a steroid hormone, which is a lipid-soluble hormone secreted in the blood, it needs to be carried by a transport globulin to get to its destination, or else it'll just sit there as a little blob of lipid soluble material that really won't go anywhere. All right. Immunoglobulins are made by B lymphocytes. All right. We'll learn about those a little bit later. They're basically magic bullets that target antigens, surface molecules on alien tissue, whether it be a single celled or a multicellular pathogen, and target them for destruction. But we'll get more into that a little bit later. Clotting factors made by the liver, okay? There's a whole host of them, okay? And they are an example of something called an extracellular cascade. We'll get into that a little bit later. That extracellular cascade, the purpose is to stop you from bleeding. And that process is called hemostasis, all right? The cellular components include the erythrocytes, which are the most abundant. They're basically bags of hemoglobin and an enzyme called carbonic anhydrase, and they're coated with a protein on the inside of the red blood cell called spectrin, okay, which helps to maintain the integrity of the red blood cell. They carry oxygen, all right? The hemoglobin is the oxygen binding pigment in blood. We have to do that because oxygen doesn't dissolve very well in water. It's just a few percent. Remember that oxygen is a non-polar gas, all right? So in order to grip enough of it to get it to the tissues, we need a special transport protein, and that's what hemoglobin is, all right? They're biconcave, so they have like a thick rim and a depressed middle. That's to increase surface area for diffusion, okay? They can change their shape as they squeeze through capillaries, like a stack of coins going from one side of the capillary bed to the other. And because they don't have any nuclear components, they don't have any biosynthetic machinery, they only last 120 days and then they have to be recycled. All right. Most of the damage that they incur comes from squeezing through these capillary beds. So after a few trips through those, they've acquired enough damage that they need to be broken down and recycled. Now, with blood, all right, one of the things that we keep, all right, is the iron. We keep the protein. We keep the lipids. Okay. The only thing that we really pitch is something called the heme. Okay. And the heme is just it's a derivative of an amino acid called histidine. And looks like a spider web. And in the middle of it is the iron atom. All right. And then the iron is what binds the oxygen. The heme itself is broken down and is excreted from the body as urochrome. That's one of the reasons your pee is yellow. It's excreted as stercobilin in your poop, which is brown. All right. And it's excreted as bile from the liver into the gallbladder, okay, which is green. All right. And that's how we get rid of it. If the liver isn't working, all right, the bilirubin, which is green, builds up in the body and turns the skin yellow and the sclera of the eye yellow. And that's a condition I'm sure you've heard of called jaundice. All right. And that indicates there's some sort of problem with the liver. Jaundice can be obstructive, and it can also be the result of liver damage, such as in cirrhosis or in hepatitis. Okay. The cellular components are the white blood cells. The leukocyte's job is to defend against disease. Okay. And you can split them into the granulocytes and the agranulocytes. All right. The granulocytes have granules that stain with something called Wright's stain, which is basically a combination of a red dye called eosin and a blue dye called methylene blue. All right. So when you do a blood smear, you take a drop of blood, you put it on the slide, you dry it, and then you hit it with Wright stain and you rinse it and then you look at it. All right. And the colors of these granules indicate the type of dye in the Wright stain they pick up. Okay, so the eosinophils pick up the red dye eosin, the basophils pick up the basic dye methylene blue, and the neutrophils pick up the methylene blue and the eosin, and that's why the granules look pink. So that's where the names came from. But what's the significance of the granules? Why do we care about them? They have enzymes that can wipe out the enemy, right? So what happens when these guys get near the enemy is they degranulate, they spray, and what they spray on their target kills it, okay? They're also capable of amoeboid movement, which means they can move from place to place, right? The neutrophils get the name polys because their nuclei have 3 or more lobes and can take on a variety of different shapes. And their job is to basically attack bacteria, okay? So they're there in early inflammation. Dead neutrophils in exudate is what forms pus, okay? Eosinophils' job, they have the red granules, is to attack parasites and to turn off inflammation, okay? They can be induced as an allergic response in if you have an allergy or something. Okay, the IgE antibody is one of the things that can promote the action of eosinophils because one of their jobs is to eat antigen antibody complexes. Okay, they attack parasitic worms and protozoa. That's another one of their big jobs. So if you have a jacked up eosinophil count, you might have a parasitic infection or you might be undergoing an allergic response. And then basophils are basically wandering mast cells. All right. And what they have are granules that stain purple and they're packed with histamine and heparin. And the job there is to promote inflammation. All right. That's their big job. The agranulocytes are the monocytes, which become macrophages when they hit the blood. Okay. And what they do is they crawl through the body looking for alien cells. And when they find them, they eat them. And when they eat them, they chop them into little bits by forming a phagolysosome. They do it by basically phagocytosis. And then they take those little bits and display them on their surface along with a protein called MHC2. All right. And what that is basically a display for the lymphocytes that recognize it to divide and make a little army of B and T lymphocytes to go out and kill whatever that little particle of the pathogen is attached to. Okay. So the monocytes become the macrophages once they hit the blood, and then they wander around the body doing their work. Now, there are some macrophages that stay in particular organs. Okay. In the liver, Kupffer cells stay there and don't move. In the lungs, there's macrophages just wander through lung tissue and don't move. And in the central nervous system, there's microglia. Okay, and in the skin, there's dendritic cells. Those are what are called fixed macrophages, right? They stay stapled to where they're supposed to be at. But these guys that are in the blood, they can go from place to place. And when they do, they can report back to lymph nodes or to the spleen or wherever and provide their display and activate the lymphocytes that recognize that display. The lymphocytes become T cells and B cells. The B cells will mature into plasma cells that make antibody when they're fully developed. The T cells will split into a variety of different subclasses. Okay. There's helper T's. Let's list these because this is important. Ba, ba, ba. Boop. And boop. There we go. So T cells. There's helper T's. There's cytotoxic. Okay. And then there's regulatory. Okay. The helper T stimulates the B cells to become plasma cells and stimulates the cytotoxic Ts to mature and show up at infected cells and tissues and kill them. All right. The regulatory Ts turn off our adaptive immunity. Okay, so they're like the off switch. And without the helper Ts, you don't get a good adaptive immune response. Basically, your humoral and your cell-mediated immunity are not worth talking about, and you get what are called opportunistic infections. Okay. Natural killer cells' job primarily is to destroy cancer cells. So what they do is they patrol the body, and when a cancer cell happens, they punch holes in it and kill it by spraying it with enzymes that induce apoptosis. And so everybody in this classroom, I don't want to scare you, gets cancer 2 or 3 times a day, right? So why aren't y'all dying of cancer? Because you have a functioning immune system that runs around and kills these cells before they get a chance to spread, and that's a process called metastasis, right? The platelets are just cytoplasmic fragments of megakaryocytes. So you might be asking, well, what the heck is a megakaryocyte? Well, mega means big, karyo means nucleus, and cyte means cell. So this is a cell with a great big nucleus. This is a weird cell that lives in the red bone marrow that undergoes mitosis but not cytokinesis. So it has this big honking nucleus, and then fragments of cytoplasmic extensions of these megakaryocytes break off, and they become the platelets that only last a few days in the blood. Okay. So we got 120 days for erythrocytes. We got 2 or 3 days for platelets, and then they got to be replaced. Right. What do? They promote hemostasis, right? They keep you from bleeding to death. They're basically bags of clotting factors that snag onto damaged tissue, release their contents, and set into motion an extracellular cascade that makes a platelet plug that is going to stop you from continuing to hemorrhage, which is a fancy word for bleed. Okay. So that's their job. That's what they're all about. A defect in hemostasis, you may or may not be aware, where you can't stop bleeding, is called hemophilia. All right. And hemophilia is basically a lack of one of these clotting factors in the cascade that causes you to continue to bleed. All right. We deal with hemophilia by giving you the missing clotting factor in order for you not to bleed to death. All right. Hemophilia is more common in men than women because it's carried on the X chromosome, which you get from your mother. And so if you get a hemophilia gene, the Y chromosome's got nothing to cover for that, and you have hemophilia. You're a bleeder. Can a woman be a hemophiliac? Yes, but it's very, very, very rare. Okay. The spleen is the largest secondary lymphoid organ, has masses of lymphoid tissue with macrophages, lymphocytes, and lymphoid follicles. Basically, what's in there are lymphocytes and macrophages, and the job is to remove pathogens from the blood itself, right? Venous sinuses are where phagocytosis of dead blood cells takes place. It stores about 300 ml of blood, so it's a blood reservoir. But basically, it's there to clean up the blood, all right, and to help recycle the red blood cells that wear out. Okay, so the liver and the spleen, and of course the GI and the kidneys all participate in this, and we'll see how a little bit later. The lymph node's job is to recover that 15% of fluid that's left behind the capillary beds that isn't picked up by the blood. That fluid enters the capillaries, then goes into the lymphatic vessels, and then eventually is going to end up in the lymph node. A picture of a lymph node is shown here. All right. And what you'll notice is that there are more incoming vessels, right? Let's get this up. These are all incoming vessels. Notice the arrow in the direction, right? And there are outgoing vessels. So the lymph from the tissue fluid flows into there. And then what's in there? Well, lymphocytes and macrophages, right? And what they do is they deal with whatever pathogens are in the lymph, and then they pass that filtered stuff out through these exit vessels. There's only a few of them. So the idea here is to slow the flow enough to give the white blood cells a chance to do their job so that you can clean up that lymph and be happy and healthy and alert the immune system to anything weird that's gotten inside your body that could cause disease. Okay. Where are these lymph nodes? Well, I'm sure you know from A&P, but they're concentrated primarily in the cervical, axillary, and inguinal regions. Okay. And you've also got quite a few in the thoracic cavity and in the abdominal pelvic.
Mr. Richard, did you say anal? Is that anal, cervical, axillary, and anal?
Inguinal.
Oh, geez.
Groin, in the groin. Yeah.
Okay.
Yeah. Abdominal pelvic cavity. All right. And the reason that they're there in the neck, in the armpit, and in your groin, right, is those are all areas where there's ports of entry, right? Cervical in your neck, look at all the holes in your head, right? You got your ear canal, you got your nasal passages, you got your oral cavity, you got your orbits for your eyes, right? And those are all places where pathogens can get inside. So you put your drainage, which is what these lymph nodes basically are dealing with, where it's going to do the most good, where it's most likely to come into contact with bad actors and knock them out. Axillary lymph nodes, why are they a lot in your armpit? Well, your lungs are right next door. All right. Your lungs are exposed to the outside world because you breathe air that's not sterile. Right. And then cells inside the lungs die. It's normal tissue turnover, and all that lymph drainage goes to the axillaries. In addition, your nipples are open to the outside world, and so that's a port of entry for pathogens as well. And then the inguinal down in the groin, well, y'all know we got holes down there, right? Everybody's got a urethral opening and an anal opening, right? And then, of course, women have a vaginal opening, and those are all ports of entry for pathogens. So you put the lymph nodes where they're going to do the most good. Why are there so many in the chest, thoracic, your lungs, okay? Abdominal, pelvic, why so many there? Your gastrointestinal system, which is open at two ends, at the oral cavity and at the anus, right? And those are all places where bad guys can get inside your body, right? There's relatively few in your limbs, okay? Because generally those aren't open to the outside world. This is also why we worry about cancer in these regions, okay? We worry about cancer in the reproductive organs. We worry about breast cancer. We worry about cancer in the neck or in the head, because a lymph node for a cancer cell can be a way to get all over the body. A cancer cell can jump from where it's formed, which is its primary tumor, into a lymph node. And then if it doesn't get dealt with there, the lymph node becomes cancerous and the cells spread through the lymph to anywhere they want to go. And that's metastasis. So this is why, for instance, if women have breast cancer, all right, one of the things they may do is remove the axillary lymph nodes. All right? Now, when you do that, you lose the drainage from the cancerous lymph node that was taken out, and you may get edema. All right, you may get swelling of everything upstream of where you pulled the node. Okay, in which case we have to put pressure garments on you to push that fluid back into the remaining drainage so that we don't impede circulation. Okay, but when your lymph node is working hard and trying to deal with an infection, it may swell up and get sore, and that's called lymphadenopathy. And that's why sometimes when you go to the doctor, he feels under your lower jaw by your neck, or he'll feel under your armpit to feel for a swollen, inflamed lymph node. All right. And that tells him maybe where the infection is at. All right. All right. Moving on. The mononuclear phagocyte system consists of monocytes that differentiate without dividing and stay in the tissues for months or years. Cells here ingest and destroy microbes and foreign material. All right. And they're able to move from compartment to compartment as needed. Okay. One of the things we're going to find out when we talk about our adaptive immunity is that in adaptive immunity, okay, you form memory cells. Okay. So those are memory B's and T's, and they stay with you for decades. Okay. Decades, let's see. And in the event they're needed, they differentiate and then provide us with all the effector cells we need to deal with the infection very quickly. This is one of the reasons why when you get sick with a particular disease and you recover and you generate memory for it, the likelihood that you'll get sick from that exact same strain of pathogen again is almost zero, because you got these memory cells waiting to jump in the event that he shows his face again. Okay. So that's the idea, right? So you get all this memory, you build it up over time. Your immune system gets seasoned to the point where you get less sick the more experienced your immune system becomes. Okay? The multicellular phagocyte system is that arm of immunity that eats the bad guys and shows them to these memory cells and also to your naive B's and T's to get them activated. Okay. This is also why vaccines work, right? Because you get exposed to an antigen you've never seen before, and then you develop memory cells to what's in the vaccine. And then if the pathogen has the same antigen on its surface, you're able to knock it out before it makes you sick. Okay. But remember that vaccines are most effective if the target of the vaccine has a low mutation rate. All right. If the target of the vaccine has a high mutation rate, the vaccine is not going to be as effective. That's why, for instance, there's no vaccine for the common cold. There's no vaccine for AIDS, okay, because they mutate too fast. But there is, for instance, a vaccine for polio because the mutation rate is very low, okay. Now, hematopoiesis is blood cell production. That happens in the red marrow in adults. Now, where is your red marrow? It's in your axial skeleton, and it's in the proximal heads of your humerus and your femur, okay? And it's working to make all the formed elements of blood every day, okay? When you're born, your red marrow is basically your whole skeleton, okay? And then your long bones convert to yellow marrow as you mature, okay? Blood cell production in tissues other than marrow, such as spleen and liver, can be a sign of disease, right? The bone marrow is one of those things that, it produces all the formed elements, but we're going to find out in the next chapter that it can go a little bit crazy and, for instance, develop cancer. And that's what produces conditions like polycythemia and leukemia, okay? And thrombocytopenia. We'll learn about all those. And sometimes what we have to do as a last ditch effort to save your life is to replace your bone marrow with donor marrow that's compatible so that we can save your life. And that's usually the last thing we try, okay? Because what will happen is that marrow will repopulate those compartments, and then you'll have basically a new source of blood, okay? What are some of the niches in marrow? Well, mesenchymal stem cells develop into osteoclasts, fibroblasts, and adipocytes, okay? While hematopoietic stem cells become the formed elements of blood, okay? And they're triggered to do that by what are called colony stimulating factors, which promotes a process called differentiation. Okay, so let's write that down. Okay. Differentiation, which is basically changing the characteristics to become the adult version of the cell. Okay. Erythrocytes are derived from erythroblasts, which are sometimes referred to as normoblasts. Maturation is stimulated by a hormone called erythropoietin, sometimes abbreviated EPO. Okay. It's made by the kidneys. So your kidneys control your hematocrit. Erythropoietin causes an increase in red cell production and release from marrow. We start out with an uncommitted pluripotent stem cell. Pluripotent. There's another word. Okay. What does that mean? It means as many possible fates. Could become many things. And then a committed proerythroblast. Pro means before. Then a normoblast, then a reticulocyte. Right. Then the nucleus is kicked out before the reticulocyte forms, and then it becomes a full-blown erythrocyte. So one of the ways that we can determine the rate of erythropoiesis is to do what's called a reticulocyte count in a process known as a differential or a complete blood count. Okay. So the more reticulocytes there are, the more rapidly you're making blood. In each step, the quantity of the hemoglobin increases and the nucleus shrinks in size. And the reason that there is no cellular machinery in an erythrocyte is to make room for the hemoglobin in order to carry as much oxygen as possible. Right? Numbers of circulating red cells in healthy individuals remain relatively constant. The peritubular cells of the kidney are what make the erythropoietin, and the hypoxia stimulates the production and the release of erythropoietin. Okay, so a little bit on this, which is kind of interesting. You may know the name Lance Armstrong. We were so proud of Lance. He won the Tour de France many, many years in a row. He was a great cyclist. Okay, and then we found out that he was cheating because he was taking erythropoietin through a needle. And what that did was that upped his hematocrit so that he could pedal longer and faster than the people that weren't doing that. And the result of that was he ended up getting stripped of his medals. And then he developed cancer all over his body. He was stage 4. And everybody thought he was going to die. And then it went into remission. And he's still with us today. Put a question mark there about why that happened. But one of the possible reasons is that he stopped using erythropoietin. Okay? Because cancer, just like any other tissue, needs a blood supply, right? And if you've got all this extra blood, that maybe helps the cancer take off and do its thing. And if you stop, maybe you starve those tumor cells and they die off. That's in fact one of the ways that we try to attack cancer is by going after, giving drugs that go after the vascularization of the tumor and starve it of its blood supply, and it dies. And it works in some cases. That's part of the mystery. Now, in professional sports, you're not allowed to do this. Okay, you can't take EPO through a needle, and you can't blood dope, which is to donate blood and save it, and then right before your event, transfuse it back into you and jack up your hematocrit artificially, right? Because that gives you an unfair advantage over everybody else. But what you can do is you can train at high altitudes. And that produces a condition called secondary polycythemia. What the heck is that? That's elevated crit. And the reason that this works is because when you train at high altitudes, your erythropoietin production goes up and your crit goes up. And athletes that do this include boxers, okay? People in the NFL do this. People in the NBA do this. People that play soccer do this, okay? I mean, it's one of those things that is going to give you an advantage over somebody that works and trains at sea level, right? And so, you know, if you're willing to do that, you're going to gain an advantage for sure because you're going to have more oxygen carrying capacity and more endurance than anybody else, okay? That's not doing that. Hemoglobin is the oxygen carrying pigment of blood, and it's made up of protein, heme, and iron, okay? The 2 polypeptide chains are the globins. There's 2 alphas and 2 betas in adult hemoglobin, each with a heme and an iron atom complex with them. The heme is brightly colored because it has conjugated double bonds. That's one of the properties of organic molecules with conjugated double bonds. And it also carries an iron atom. And turns out that metal ions are brightly colored as well. So that's why your blood is red, okay? The heme and the globin and the iron are perfectly suited to pick up oxygen in the lungs and dump off oxygen at the tissues. Otherwise, we wouldn't have enough oxygen dissolved in plasma to keep us alive, okay? So the idea here, right, is that under conditions of, let's say, normal pH, normal body temperature, okay, and high oxygen concentration and low CO2 concentration. Hemoglobin, HB, that big H little b, picks up oxygen. Easily. Okay. And these are the conditions in the lungs. Okay. But at low pH, high temperature, low oxygen, and high CO2, okay, hemoglobin unloads its oxygen. And those are the conditions that exist in your tissues. So this is the perfect protein to do this job. Okay. Otherwise, we wouldn't be able to carry enough oxygen in the body to keep us alive. And remember, you make 20 times more ATP with oxygen than without it. So we are what a biologist would call an obligate aerobe. We have to breathe oxygen in order to live, right? Oxygen is about 20% of the air that we breathe. And thank God for hemoglobin to grab that 20% out of the lungs and get it to our tissues so that we can make ATP and survive. Okay. What do we need to make hemoglobin? Well, you need protein. You eat protein to make protein. You need vitamins. You need B12, B6, B2, vitamin E and C, and folate, and pantothenic acid, and niacin. You get all that in your diet. Okay. And of course, you need iron. And you also need copper. Okay. It's part of the enzymes that help to manufacture the hemoglobin. All right. And then when you're ready to break the hemoglobin down, right, when you're like recycling red blood cells, you go through a process like this. So let's look at this here.
When old red blood cells rupture, the released hemoglobin is ingested by macrophages. The globin chains of hemoglobin are broken down to individual amino.
Hold on a second. Cut off like that. We'll get it. This is going to involve your liver and your spleen. The things about iron that is unique is that iron tends to form salts in with other nonpolar molecules, or sorry, other nonmetals that don't dissolve very well in water. Okay, an example would be iron oxide, which is rust. Okay, so we try to, whenever we carry iron around, we try to complex it with a protein that keeps it away from other anions, so that it doesn't end up becoming insoluble. Okay, so those things like hemosiderin and transferrin and stuff like that. Leukocytes, the development of leukocytes is called leukopoiesis, right? They come from stem cells in bone marrow. The granulocytes mature in red marrow. The agranulocytes and the monocytes are released in the blood before they're fully mature. Growth factors and CSFs encourage production and maturation of white blood cells. Production increases in infection and in the presence of steroids and reduced bone marrow reserves. Okay. One of the things that we should note about this, all right, is that the lymphocyte population has a very particular sequence of events, right? The B lymphocytes, M to M, and monocytes, and granulocytes, all mature in bone marrow, right? And then are released into the blood. Okay. But the T lymphocytes are a little different. So they start in the bone marrow, in the red marrow, and then they move to the thymus and are then released. Okay. Hold on. I'll mute it. There we go. And are then released. Okay. So that's why they're called T lymphocytes. They mature in the thymus. The B lymphocytes mature in the bone marrow. B for bone marrow, T for thymus. Okay. All right. Development of platelets comes from megakaryocytes, right? The megakaryocyte undergoes mitosis, but not cytokinesis. So it gets a huge nucleus and it has these cytoplasmic extensions that extend into sinuses in the red marrow. And then those fragments that break off, they become platelets. Okay. Platelet levels are regulated by thrombopoietin. They circulate for about 10 days before losing functional capacity. Senescent platelets are sequestered and destroyed in the spleen. So there's our spleen again, right? Hemostasis just means the cessation of bleeding, right? Requirements include the production of a platelet plug and then a fibrin clot. And then finally, the dissolving of the fibrin clot after tissue repair has taken place underneath it, okay? It involves an extracellular cascade of clotting factors that are made by the liver and are always in the plasma, okay? As well as responses of the blood vessels, okay? The vasculature will constrict to reduce flow, and that promotes the clotting cascade as well as the platelets being trapped on damaged tissue and releasing factors that attract more platelets. And that's another example of a positive feedback loop, okay? So let's take a look at hemostasis.
Hi everyone. I want to start off by talking about Mary. Mary likes to play soccer, and she was at one of her games and she was running really fast. She scored a goal, and then she tripped and fell and scraped her knee. And then she started to bleed. Now, normally our bodies want to make sure that our blood, which it flows through our blood vessels, stays in our body. But if we have an injury that allows blood to leak out of the blood vessel and out of our body, we want to make sure we can stop that. And we do that through a process called hemostasis. And it's through hemostasis that we create this clot. And this clot allows the bleeding to stop. It stabilizes the injury in the blood vessel, and it gives the blood vessel time to heal. Now let's take a look at exactly what's going on in Mary's knee and exactly how we make this clot. So here we have a blood vessel, and in our blood vessel, we have our blood. In our blood, we have many different types of cells. One of the most important ones is our red blood cell that carries oxygen all over to all our tissues in our body. And in this blood vessel, we make sure that blood is flowing smoothly in one direction. Did you know that if we were to line up all of our blood vessels, the total length of our blood vessels would come out to about 150,000 kilometers, which is about 93,000 miles. This is the same distance as if we were to travel around the Earth a little bit less than 4 times. So you can see we have a lot of work, our blood vessels have a lot of work to do. And one of the cells that helps with this are the cells that line the inner wall of the blood vessel, the cells that communicate with the blood. These cells are called endothelial cells. And it's these cells that allow the blood to flow smoothly. And these are the cells that communicate with the blood whenever there's been an injury and hemostasis needs to begin. Now let's take a look at Mary's knee. She scraped her knee, so let's cause a break in her skin. Let's say this is her skin. And she also caused damage to the blood vessel underneath. So let me erase this. Let me cause some damage to the blood vessel. And so you can see blood that should be flowing in this direction now can come out of the blood vessel and out of the skin in this direction. We want to stop this. And the way we stop this is through hemostasis. There are 2 stages to hemostasis. The first stage is primary hemostasis. And this begins immediately after there's been an injury. And what happens is the endothelial cells at that site of injury begin to secrete proteins. And this sends signals to the blood that there's been an injury. And in our blood, we have platelets. These platelets are made in the bone marrow, just like red blood cells. And their purpose is to respond to injured blood vessels. And I drew them as squares, but they're actually about this small compared to a red blood cell. The reason I drew squares is I want to make a note of what they carry inside of them. They carry these sacs or granules that carry molecules and proteins that are necessary for hemostasis. So they store them and they're ready for use whenever needed. And so what happens is the endothelial cells secrete these proteins and send a signal to the platelets to come to that site of injury. And then a sequence of events happens with the platelets that I won't go into details here, but it allows the platelets to stick to that site of injury. They get activated, they change shape, they release what they have inside, and then they start clumping together and call more platelets and cause more platelets to clump there as well. And what we end up with is what we call a platelet plug. And this is the first step of stopping any bleeding. But this is still kind of weak. And we need to make this stronger. And that's where secondary hemostasis comes into play. In secondary hemostasis, we make this platelet plug stronger with a protein called fibrin. And what fibrin does is it lays over and links up on top of this platelet plug and makes it tighter and sturdier. It's like tying a knot. In order to prevent it from getting loose, you double knot it or triple knot it. And this is exactly what fibrin is doing. But fibrin doesn't flow in our blood as fibrin. It carries around an extra piece of protein. And when it has this extra piece of protein, it's called fibrinogen. So we need a way of getting fibrin from fibrinogen. And the way we do that is with this family of proteins called coagulation factors. And I'm drawing them all in one color, but there are several different types of coagulation factors. And the injured blood vessel will send signals to these coagulation factors to get activated. And once one gets activated, it will activate another, and activate another, and activate another. And then ultimately, that extra protein on fibrinogen will get cut off, and that will allow fibrin to link up on the platelet plug and make it stronger. This sequence of activation of coagulation factors is what we call the coagulation cascade. I won't go into the details here either, but it's just key to remember that in secondary hemostasis, we make this platelet plug stronger by getting fibrin from fibrinogen through the coagulation cascade. And it's with this fibrin mesh over the platelet plug that we create this clot. And it's this clot that stops bleeding, allows blood flow to continue through the blood vessel, and allows the blood vessel to heal.
So with platelet activation, we latch on to damaged tissue, we degranulate, we generate a platelet plug because these factors attract more platelets, okay? And in addition, we have vascular spasm, right? So the smooth muscle in the blood vessel will clamp down a bit, and that'll slow flow, and that concentrates clotting factors, and that promotes the clotting cascade, okay? And eventually what you're going to generate is a fibrin mesh that's going to trap other formed elements and allow basically to close the opening so that the bleeding will stop and then set the stage for tissue repair underneath, right? There's a variety of pathways that produce a fibrin clot. The intrinsic pathway is activated when factor 12 comes into contact with subendothelial material exposed by injury, while the extrinsic is activated when tissue factor is released by damaged endothelial cells. And then there's a common pathway as well. So let's take a closer look at this.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves two pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascade meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the two pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A, in combination with 5A, activates thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
So for treatment, we give the missing clotting factor so that you can form a clot, okay? Once the clot has done its job, right, we don't need it anymore. In fact, we don't want it anymore. In clot retraction, the fibrin strands basically pull the wound together so that there's less possibility of, rupturing it and bleeding again. That's aided by platelets within the clot itself. But then after we've done tissue repair, fibrinolysis takes place, right? We actually dissolve the clot. Plasminogen and plasmin help us do this. Plasminogen is the precursor. It's converted to plasmin. And then the fibrin is broken down. Okay. You can see how this works here. Okay. TPA converts plasminogen to plasmin, breaks down the fibrin, and you've got new tissue underneath. So you don't have to worry about it at all. Right. The vascular endothelium has a role in this as well with UPA and UPAR. Okay. Basically at that point we're done. This is critical, right? Because if the fibrin clot were to stay there and dislodge, then it could become an embolus. Okay. Blood cell counts increase above adult levels at birth because of the trauma of birth and cutting the umbilicus. Hypoxic intrauterine environment is going to stimulate erythropoietin production, resulting in polycythemia. Okay, so here we get the definition for polycythemia. Okay, polycythemia is an elevated red blood cell count. Okay, an elevated CRIT. Okay. Now, primary polycythemia is a disease process, and that usually indicates some problem with the red bone marrow, right? But secondary polycythemia is a normal response to high altitudes or low oxygen concentration, okay? Children tend to have more atypical lymphocytes as a result of viral infections as well, okay? What are atypical lymphocytes? Those that are not frequently seen, like basophils, and in some cases, lymphocytes that may have alterations in their structure. Okay, the erythrocyte lifespan is normal, but erythrocytes are replaced more slowly as we age because there's less tissue to do the job. Bottom line, okay. Possible causes include iron depletion, decreased serum iron, iron binding capacity, and intestinal iron absorption. Lymphocyte function decreases with age as well, and the humoral immune system is just less responsive. Okay, and that's true for all our body systems as we age, basically. Okay, there's just fewer cells to do the job, so the body system is less effective. Right. All right, let's bust for 20 and give me a chance to whip up some Q&A for you, and then we'll do a little bit of chapter 21, and then we'll only have a little bit of chapter 21 to do on Wednesday, and that'll leave you guys more time to deal with the quizzes and the second attempts and everything else. Okay, so let's do that now.